So last class we completed cholinergic nervous system, that is cholinomimetics, anticholinesterase agent, that is indirectly acting cholinomimetics, anticholinergic drugs, and drugs which blocks NM receptor, that is skeletal muscle relaxant, and finally the ganglionic blockage. Now we start with adrenergic nervous system. So in case of adrenergic nervous system, there are two neurotransmitters, one operating in the autonomic ganglia of the adrenergic nervous system and one neurotransmitter which operates at the effector junction. Kindly refer back the initial classes on neurohumoral transmission. So acetylcholine is the neurotransmitter at the autonomic ganglia, but the effector junction, where the sympathetic nervous system produces an action, the effector junction, the neurotransmitter is noradrenaline, and it is synthesized from a precursor molecule, amino acid phenylalanine. But first it is hydroxylated in the liver to tyrosine, and in adrenergic neuronal cell bodies, the adrenal medulla as well as the dopaminergic neurons, the tyrosine will be taken up and converted to dihydroxyphenylalanine, that is DOPA, by the tyrosine hydroxylase enzyme present in the three locations as I mentioned. And the DOPA will be converted to dopamine by the DOPA decarboxylase enzyme, which is also present in the dopaminergic neuron, in the adrenal cell bodies, and in the adrenergic cell bodies. So the DOPA will be converted to dopamine, only up to dopamine in the dopaminergic neurons, because they don't have the other enzyme like beta hydroxylase. They lack beta hydroxylase enzyme. So if you consider the dopaminergic neuron, it will be having two enzymes, that is thyroxine hydroxylase and dopa decarboxylase enzyme. Only two enzymes in the dopaminergic cell bodies. So dopaminergic neuron will synthesize only up to dopamine. While if you consider the case of adrenergic neuronal cell bodies or adrenal medulla, they do have one more enzyme other than tyrosine and dopa decarboxylase. They have one more enzyme, that is beta hydroxylase. So in the adrenergic cell bodies and adrenal medulla, the synthesis continues, and the synthesis initial conversion of dopamine is to noradrenaline. Until that step, it happens in the adrenergic neuronal cell bodies. Why? Because adrenergic neuronal cell bodies will be having the three enzymes, tyrosine hydroxylase, dopa decarboxylase, and beta hydroxylase. So they will synthesize noradrenaline from the tyrosine, but they cannot synthesize adrenaline. They cannot synthesize adrenaline because the N-methyltransferase enzyme is absent in the adrenergic neuronal cell bodies. So the presence of the enzyme will determine till what the phenylalanine will be converted. So in the dopaminergic neuron, it will be converted only up to dopamine, while in the adrenergic neuronal cell bodies, it will be phenylalanine or tyrosine will be converted till noradrenaline. While in the adrenal medulla, there is one more enzyme available, that is N-methyltransferase. That is the reason why adrenal medulla will synthesize adrenaline and secrete adrenaline. And kindly note down, dopamine and noradrenaline, they are neurotransmitters because they are secreted from the neurons, while adrenaline is considered as a hormone because it is secreted from a gland, that is adrenal gland, from the adrenal medulla. So kindly remember the three, dopamine, noradrenaline, and adrenaline, they are known as catecholamines because of the structural similarity. But dopamine and noradrenaline, they are neurotransmitters. Adrenaline is a hormone. So how the noradrenaline is stored in the adrenergic neuron? They are usually stored in complex with ATP in the ratio 4:1, and they are usually adsorbed to a protein known as chromogranin, and they are released in response to the impulses by a calcium mediated exocytosis process. Coming to adrenergic receptors, kindly note down, it's important. There are two types, alpha and beta receptors. Alpha adrenergic receptors, there are two subtypes, alpha 1 and alpha 2, while beta, there are three subtypes, beta 1, beta 2, and beta 3, depending upon the location where they are present. And all the adrenergic receptors, they are G protein coupled receptors, that is type 2 receptors. Coming to the receptor, their location and the action produced by the activation of the receptor by an agonist or the endogenous signal molecule, that is noradrenaline. I repeat, the action on activation means it is the action produced by the activation of the receptor either by the agonist, maybe a drug, or by the endogenous signal molecule. The endogenous signal molecule, there are two signal molecules which can activate the adrenergic receptors. One, it is the neurotransmitter noradrenaline, and second one, it is the hormone adrenaline. So let us see what are the receptors and location and their actions. First one is beta 1 receptor, which are located in the heart. So usually, as you know, adrenaline produces cardiac stimulation. So the cardiac stimulation of adrenaline or the sympathetic stimulation of heart is done through beta 1 receptor located in the heart. Second location of beta 1 receptor is in the JG cells, that is juxtaglomerular cells in the nephron, and their main role is to secrete renin. So adrenaline, if it is secreted in response to fight or flight, or if there is a sympathetic stimulation, it will cause the release of renin, R-E-N-I-N, renin, resulting in activation of renin angiotensin aldosterone system, resulting in increase in blood volume and rise in blood pressure. Second, beta 2 receptor, important receptor located on smooth muscles, and there produces relaxation. Bronchial smooth muscle, it produces bronchodilation, useful in asthma treatment. In vascular smooth muscles, in the blood vessels, it produces vascular smooth muscle. It's a blood vessel is missing there. Can you note down? It produces vasodilatation. In the eye, the blood vessels in the ciliary processes will undergo vasodilatation. As a result, there will be increased secretion of aqueous humor and increase in intraocular tension. So this can be blocked by blocking beta 2 receptor in the eye and can be used for treating glaucoma. I repeat, the beta 2 receptor, if it is blocked in the ciliary processes of the blood vessels, it will decrease the secretion of aqueous humor and can be used for treating glaucoma. In the uterus, that is gravid uterus, meaning the pregnant uterus. So only the uterus, when it is pregnant, it will be responding to the sympathetic nervous system, and it will lead to relaxation of the uterus, and the effect is known as tocolytic effect. I repeat, tocolytic effect. So the relaxation of uterus is performed in order to prevent the threatened abortions or in case of prematurity and so on. Liver and skeletal muscle, the beta 2 receptors promotes glycogenolysis and increases the blood glucose level. In GIT and urinary system, it produces relaxation and decreases the urine output. Beta 3 receptor, it is located in the adipose tissue and promote lipolysis. As a result, there will be increase in free fatty acid level in the blood, which can be used for gluconeogenesis and again increases the blood glucose level. Coming to alpha 1 receptor, they are located on the smooth muscles and produces contraction. See the difference? There are two receptors, beta 2 and alpha 1. Both are belonging to the same system, adrenergic system, but produces opposite effect. So in the blood vessels, both beta 2 and alpha 1 receptors are present in the vascular smooth muscles. Beta 2 receptor in the vascular smooth muscle, if it is stimulated, it will lead to bronchodilation. While alpha 1 receptor in the vascular smooth muscle, if it is stimulated, it will lead to vasoconstriction. So that means the adrenergic nervous system alone controls the tone of the blood vessels. Through beta 2 or alpha 1, they maintain the diameter of the blood vessel, that is the tone of the blood vessel, what you call vasomotor tone. The liver, it promote again glycogenolysis. In the eye, radial muscles of iris, already mentioned under parasympathetic nervous system. Eye, there are two muscles, that is circular muscles of iris and radial muscles of iris, which controls the size of the pupil. So radial muscles of iris, if it is contracts, it will result in increase in pupil size, leading to mydriasis. Can you note down there? It's missing there. Eye, it causes contraction leading to mydriasis. The ciliary processes, it will decrease aqueous humor secretion, because there will be vasoconstriction. Beta 2 receptor, it was vasodilatation, so that lead to increased secretion of aqueous humor, while ciliary process alpha 1 receptor will produce vasoconstriction and decreases the secretion of aqueous humor and decrease in intraocular tension. So the drug which stimulate alpha-1 receptor can be used for treating glaucoma. The male sex organ, it is involved in the process of contraction of the organs associated with ejaculation and promote ejaculation process. Nasal mucosa, the stimulation of alpha-1 receptor will produce vasoconstriction and decrease the thickness of the nasal mucosa, and it is useful in case of treating nasal congestion. Alpha-2 receptor, it is also known as autoreceptors. They are located both pre and post junctionally in the brain, and if that receptor is stimulated, it will decrease the sympathetic outflow from the brain. That means it decreases the sympathetic activity from the brain to the peripheral organs. Remember, when the receptors are stimulated, they are supposed to produce a positive response and action. But here, alpha-2 receptor is an autoreceptor located in the brain. If it is stimulated, if it is activated, it exerts a negative effect. It will decrease the sympathetic activity from the brain. But if it is blocked, it will be producing increased sympathetic activity from the brain. Other location, they are also located in pancreatic beta cells and decrease insulin release. In platelets, they promote platelet aggregation. Thank you very much. Study well.